he doesn't want to hold it. For that.
Okay, we're just gonna start and we'll see how it goes. And I'm happy to use the mic. Okay. Hey guys, my name is James. I'm the former president of the Pikes Peak Mycological Society. And I've been studying mushrooms a long time in Colorado, probably about 2 years, if I count. And somehow I got involved with Agaricus at one point, and then I got obsessed and then ended up here. So we have a lot of stuff to talk about today, and I'll probably go through pretty quickly some of the slides. But feel free to ask any questions in the middle, and we'll try to answer those questions, and then we'll answer more at the end. This is a project that I started 2001, 2002, something like that. Started with 12 scientific collections and now have over about 150. So it's grown considerably, and I've had a lot of help from a lot of a lot of mycologists have helped me get to this point and brought me the tools that I needed and kind of pointed me in the right direction. So there are a lot of people that I can thank. But before we get started, I want to talk about the book project that we were a part of with both clubs. This actually got me really hyper-focused on Agaricus when I committed to writing this section here. And I started thinking about it in different ways. I started getting more detail. They started doing, more measurements. And next thing you know, it just kind of exploded. So I'm really proud of the book we did. It's great for beginners. Highly recommend it. And then proceeds of this book go to both clubs to help with future projects that we're doing like this. So.
Sell that at our commission.
I would ask Alexis. Probably.
Well, they always, but I've never seen that one on ours.
Really? All right. So let's talk about, remove this. What is an agaricus? So, most people think an agaricus is the button mushroom you find at the grocery store, or a mushroom in general. When you think of mushroom, this is the first thing you think, unless you're thinking about Amanita muscaria, most likely. They can be difficult to identify in the field. And so, there are a lot of little tools that we use to identify them, and I'm going to show you some of that stuff today. Agaricus bisporus is an interesting one because it's, at the grocery store, and, they come in different colors. So these are the same species. So the carini and the white button mushroom are actually the same thing. They're just a different strain or kind of different version of it. But if you were to DNA sequence these, they would come out to be the same thing. Same thing with the portobellos. The portobello is essentially a larger carini that's been opened. And that's what most people know as agaricus. But in Colorado, we're at like 47, 48 species that we've counted. And we're probably going to go over 50. And there's certainly more that we haven't found. And a lot of the work that I've put into this has data. A lot of it's stuff I've collected, but then we also have pictures and data from other people as well. And so anyone who's using iNaturalist, as Alexis was promoting, if you take pictures of any of these agaricus or any mushrooms in general and you put them up there, you're helping other citizen scientists with data, especially if you're sending them out for DNA sequencing. So, I've managed about 150 or so collections, and they look like this. Right here is Agaricus arvensis, dry collection. This has been
James.
Yes.
I'm sorry to interrupt, but the volume is really fluctuating in and out, and sometimes I can hear you great, and then sometimes it sounds like a deep, echo kind of thing.
Okay, thank you, Catherine.
Oh, and that sounds good now. Thank you, James.
It's probably because I'm walking away from the microphone. I'm not used to using a microphone, so I might have to use it. But thank you for your input. So anyway, if you collect collections and you dry them and you send them for DNA sequencing, and you send a little piece and you keep the collection, you could eventually send this to the Denver Botanic Gardens, and it can be stored for science forever. And then people who do future work, they can come and pull it out and do work on it. They can do microscopy, they can sequence it again, they can do all sorts of things. So these scientific collections are very important, and we're losing, certain herbariums like the Duke herbarium, and it's really unfortunate that the funding is gone. And so DBG houses thousands of collections, and it's really a big deal. But it's really important that we do collect stuff and take pictures of it and document where it is. And it's not just about the DNA, it's also about where you find it, Um, it provides a ton of data for fungal conservation purposes. And in time, it will generate more projects and funding for preserving the fungi that we do have. Anyway, so Agaricus are saprobic. So what they do is they decompose dead organic matter, and that's mostly pine needles and dead grasses and things like that. They're not mycorrhizal, even though they do associate with certain trees. Oftentimes, you'll find certain Agaricus under spruce most of the time or something like that. And that's because they do have some sort of relationship that we don't know, but there's something going on there. They're just not necessarily wrapping around the roots like a bully would or something like that. We have about 500 to 600 species that are described worldwide, and there are a lot more that are undescribed. We know it's over 500, so I put 500 to 600 because we know it's within that range currently. So what is an agaricus still? So the flesh can be staining yellow or not stained at all, orange, pink, red, a lot of different colors, and that's one of the ways you can discern the different ones. So staining reactions are a big deal. They all have a chocolate brown spore print, so really they have the same spore color. They have a stem or a stipe that breaks freely from the cap. So if you were to take one of these guys, you'll see it's kind of like a Russell almost, where it'll just crack right off and it'll break, like a piece of chalk. Cap colors can vary. You know, most of the time you see them, they're white, but often, you know, browns are pretty common. And then some of them can go yellowing, some and you know, the gills, depending on which section they're in, can start at a different color. So showing the life cycle of Agaricus campestris right here, you'll see that as soon as they open, they're pink. And that's why they were called the pink bottom, right? So it's very common that, you know, gills will be pink, especially in that particular group. So if you found like something in the Arvenses group, it wouldn't necessarily be pink. It would be like white. And here's an Sorry, you guys at Zoom can't see this, but someone brought some agaricus. And if you look at this one, this is in the button stage. And I can tell it's in the Arvenses group, but if you look at, there's a little bit of gill color there, and it's really light in color. So it won't be pink necessarily. It'll start off like white and a little bit grayish. And then it might have a little bit of pink tones and it'll go right to dark. Kind of like one of these. So this one's turning color. But with campestris or the campestris group, they will always start out pink. And so if you find mushrooms like that on a lawn and you're like, okay, well, I don't know if this is campestris or it's Arvensis or what, one of the first things you need to do is look at the gill color at that stage, if you can. Smell is obviously another thing, and we'll get into more of that. But there are little subtle differences. So where are they typically found? They're typically found in lawns and fields, and then also under conifers, pretty common. Sometimes they're with trees, sometimes they're not. We do have several that are found above tree line, which are really interesting, and we need to collect more of those. And I know when Kathy Cripps did her presentation, she talked about Agaricus chinodermis and Agaricus altipes being above tree line, which does make sense. Chinodermis is found at higher elevations. Altipes is a little more rare in Colorado. But you can also find these at your local grocery store. Not all of them, but some of them. So when we identify Agaricus, one of the things we want to look for first is, okay, where are you? So are you in the mountains somewhere at high elevation? Are you, you know, over here at the park in Denver, you know, picking something off the ground that's, you know, in the grass? Are you near trees? Are you, you know, in a riparian area? But mostly it's, you know, are you in a montane or an area with conifers up higher? Or are you in, you know, the city on the Front Range on a lawn? And many of these lawn mushrooms that we have here on the Front Range also occur in cities like Telluride on lawns. So a good example of that would be Agaricus arvensis. It generally occurs down here at the lower elevations on lawns, but it'll go up into the ponderosa pine about 7,000 feet or so, typically in June. But they can grow throughout the year anywhere with the right, you know, components or right habitat. So like in Telluride, even on a lawn that far west, you could find Agaricus arvensis, and it's been found there. Same thing with the our Xanthodermis relative. Anyway, so you want to look at the habitat. So that's really important. Habitat comes first. it's good to look at the morphology. How big is it? What does the ring look like? Does it have a bulbous base? Oftentimes. Can't see on any of these guys, but a lot of the woodland agaricus have like a bulbed base on them. And it's one of the ways that you can kind of narrow things down. Smell is a big factor. So you'll see whenever I talk about this stuff, I'm smelling these a lot. And so this one has a very unique smell that someone brought in here from the mountains. And I can tell this is like something that's in Section arvensis based on the way it smells and where it came from. So there are two things. It's also got yellowing on it. So if you look at the staining color, like we're talking here, this one is technically a yellow stainer. And I don't know if you guys can see that. I know people on Zoom can't see it, but this has a little yellowing. All right, so some of the other ways we identify on top of that, obviously is to do microscopy. You can see there's an example of that here. So this is Agaricus brandeli, which is related to Campestris. And one of the bigger things with spore measurements in Agaricus and identification is the spore length. A lot of them have a different spore length. They also are shaped a little bit different, and sometimes they have what's called an apiculus, which these don't, which is like a little point on the end of them. So they could be a little bit different. Another thing that we use, obviously, is a DNA barcode, and this is just the ITS region, which is a very small region of the DNA. And that's the most common region of the DNA that is compared, between different fungal species. There are other regions too, but this is basically like the easiest thing to find other mushrooms to compare to. Here's a PPMS foray right here. You can see the different Agaricus here, and what happened was I got up to run this foray that morning. I had some Agaricus bernardii from the day before in this little bin right here. I looked out my backyard and then I saw these Agaricus when I was looking out from the sink, getting my coffee, and I went, wait a minute. And I went and I grabbed those. And then I think I found these other ones over here on the right the day before. So I went, hey, I have 3 different Agaricus species. This is perfect. I brought them to the foray. And before we started the foray, I said, okay, who wants to learn about these? And I threw them all out on the ground and I said, all right, somebody tell me what the different ones are. Can you guess what species these all are? And I started talking about, the smell. They smell different. So everybody get a good smell and get an idea of what they smell like. So we have Agaricus arvensis over here on the left. We have bernardii here in the little bin, and then we have Bisporus on the right. And I found this picture in my pictures, and I went, this is a really good one to show because we only have our friend Jennifer Bell here in the back smiling. But this kind of shows you, one of the first places to start. Okay, where are you at? What does it smell like? Agaricus toxins. So one of the reasons you probably don't want to eat a lot of city Agaricus is because they will absorb heavy metals, especially cadmium, according to Paul Stamets in his book. I don't know how up to date that is, but I'm sure there's other things they can absorb. Agaricine has been something that has been talked about, and it's a little controversial because it's caused tumors in mice. And so it's mostly destroyed by cooking. However, there are a lot of different studies on that, and the consensus is it's not really a toxicological risk to humans, especially if you're cooking your mushrooms or letting them sit in the fridge for several days. It dissipates it. It's mostly destroyed by cooking. There are also gastrointestinal irritants. So stuff in the xanthodermis group is, some people can eat them, but most people can't. And that's really the toxic group that we have. Another thing are bacteria and other fungi, but Agaricus need bacteria to even kind of grow and fully fruit out. So if you go to the salad bar and you're eating, raw button mushrooms, just know that you're getting agaricine and you're getting probably some bacteria in your diet as well. So that's one reason not to eat raw Agaricus. I know a lot of people eat raw mushrooms, certain kinds, like truffles and possibly like really, really clean porcinis and stuff like that, which are a little bit safer. But just know that if you eat raw Agaricus, you're exposing yourself to some of these things. And it's probably not that big of a deal, but that's what's happening. So are agaricus safe to eat? I think the majority of the ones we have in Colorado are edible. And, you never eat one raw, like I was saying up here, for those reasons. The majority of the toxins are destroyed by cooking. And then, lawn agaricus, city agaricus are obviously going to have more city pollutants. There's cars driving by, brake dust, there's all sorts of things happening in the city. And there's a lot more pollution than, say, if you're up at 10,000 feet, where you're under some spruce tree and you're picking porcini and all of a sudden some agaricus pop out, right? So when I buy them, I prefer organic agaricus, and that's what I have over here. Just to kind of like have the cleanest things that I can be eating, but I much prefer the wild. And this is a picture from when David Aurora came in 2021, and we went out and found Agaricus julius, our state mushroom. And we found a couple different ones. So we have amicosis here, and then julius, and then didymus. And we did a taste test. We're like, okay, let's cook these all the same way, and let's see what they taste like. Which ones do you like the best? Well, I like julius the best. I think it's the best one. I like didymus next. And then, amicosis is a darker agaricus, and I feel like it doesn't have as much flavor, and that's my personal opinion, but it would be better paired with something like a steak. Okay, so how old are agaricus? This is a new one for me. I was doing some research, and I read this agaricus paper that blew my mind, and I was like, oh my God, this ties to the geology I've been learning and geologic timelines and things that happened millions of years ago. So what I found out was that agaricus diverged around 66 million years ago. At least that's the estimate. And if you go back and you say, what happened 66 million years ago? Asteroid hit the Gulf of Mexico, and the dinosaurs went extinct. And then all these fires, wildfires were all around the earth, and a good amount of the species on the earth went extinct. And then there was all this organic matter everywhere. And that started the great fungal bloom, because what happened was all the fungi were like, great, we have all this food. And they just started eating all this stuff that got destroyed. And then they just blew up. And definitely read more about the great fungal bloom. I think it's fascinating. So after that asteroid hit, it changed the earth completely. All the smoke from the fires covered everything. And then a lot of stuff perished, but the fungi were just exploded at that time. And agaricus, that's where they started. So they did a fossil calibrated analysis to estimate the divergence times between agaricus and its outgroup. Oops. And they estimated at 66 million years old, which I think is fascinating because of what happened at the Earth at that time. And if you think about what Colorado was like at that time, about 70 million years ago, Colorado was actually an ocean for the third time, at least on this side of Colorado. It was a shallow ocean. And then after that, the land started rising, and it completely changed. And it was actually subtropical around that time. And Colorado has moved to the north considerably. And Colorado was actually around the equator. That's from what I understand. Oh, one more thing. So I thought it's fascinating. You can see the different clades or the different sections over here, and you can see the millions of years ago when they split. So you have the main section, the Arvense section, Menoris, and then there's all these other ones. And so these are the estimates of when they kind of branched out from each other. And I thought that's really super cool. I do not know how accurate that really is, but that's the estimation. So agaricus are 66 million years old, and we had a bunch of them split, about 30 million years ago. So this is kind of like my map of agaricus of Colorado. This is what I had been working on to figure out, what species we have. I started with 12 collections, not thinking about how many this would blow up into. I just was like, okay, I find some agaricus I can't identify. So if you have, Vera's book, she has several agaricus in here, and I learned all these common ones, early on. So Arvensis, Bernardii, all the stuff is in this book. Campestris, Amicosis, and to me, all the ones that are in the easy ones to learn to identify. They're easy to differentiate, but as we collected more data, we started finding all sorts of different things, and a lot of this stuff looks, the same. And that's something where people often ask, okay, what is going on with this group in section arvenses? Well, there's a ton of different ones. There's probably like 12 different ones that we're aware of. And a lot of them look the same, except for the Julius and Augustus group here. The majority of these all look almost identical, or a lot of them do. They're slightly different. So there's no way in the field you're going to tell the difference between Agaricus didymus. They look identical. They're so closely related. They could be the same thing. And then Semensis is a little bit different, but if you have Semensis in your hand and you had didymus in your hand, I'll guarantee you couldn't tell the difference. They just look, they're too closely related. So if you look in the book that we did, we called them woodland Agaricus for that reason, because there's so many woodland Agaricus that are yellow standers that are in this group. And these are all the ones that are known from Colorado. And these are all the ones that are either have been found via DNA sequencing or have been documented previously. So, you know, some of these don't have DNA sequences behind them, like Clematidopus, like we can't find that, but it's been documented here. And we just need to make more collections and hopefully sooner or later we can find it, but it may not even have a sequence. So some of these don't even have a sequence. And we think like Cuniculiacola, like that one doesn't have a type sequence. It could, but the holotype is from 1989. It's at San Francisco State University. And I'm trying to figure out if they have sequenced that, because I believe there's a possibility that this one actually is one of these other ones. So I built this, and you can see the different sections, and they're different stainers, different things going on here, and they're grouped in different groups. And that's how I like to look at them. So if you go out in the woods and you find something that's like bright red staining, like this guy right here in the Sylvaticus group, then you can narrow down kind of like which group it's in, what's it related to. But this is the list I put together of all the different ones. There's at least 47 on here, I believe, somewhere around there. And there's a couple that might not be on here, but these are the ones that we know do exist here. Most of these have DNA data. So here's another look at it. This is actually a phylogenetic tree that I learned to put together. And thank you, Amy Honan, for helping me figure out how to build phylogenetic trees properly, if you're listening. This isn't the whole tree of everything, but this is the tree of the species that are DNA verified in Colorado. So every single thing on this tree, we verified with DNA sequences. And this is from, you know, citizen science, scientific collections, not just me. This is from other people that are collecting too. People like Dimitri, who's here in our audience. He's doing a lot of collecting. And, you know, the work that he's doing helps build this stuff, because we can take that data and incorporate it within our study. And the more people doing that, the more we can learn about what we really have here. So I broke it up into sections so you can see how these are all related in the Arvense section. And if you were to cut this off right here, that basically says that these all have a common ancestor right here. And they're also very closely related to section Minoris, which are these really small looking Agaricus. And so they branched way back here. And if you've ever seen any of these and you've smelled one, they smell very similar. They have the same smells. They're yellow stainers, but they're just somewhere back in, you know, time, they split and they became really small, where the Arvenses group stayed big. So it's really interesting to see that. But I broke it up into sections, and I wanted to show this because I think it's important to see all the ones that we do know are verified. So if you go back to this, you'll see there's some names on here that may not be on that tree, and that's because we don't have the DNA data to back that up. We do. So this is all DNA back data right here, all of it.
What do the numbers mean?
So the numbers are basically, they're called bootstrap numbers. So anything with 100 is a really, like, it's almost 100% certainty that that's where it did branch. Any of the lower numbers, there's less certainty. So if you look at something that has like, you know, 8 over here, that's much less certain that it actually branched right there. That's what that means. So we want a higher number. So the higher the number, the better, but we don't control that. So when you see a number like 99 or 98 or 86, like that's pretty good. That's very likely. Here's another way to look at it. I did a circular tree. I thought it was really cool. I almost didn't put it in here, but I just kind of liked it. I thought it was the coolest tree I ever made. You know, it may not be practical for visuals, but I thought it looked cool, and I'm really happy about it. So
It's your slideshow, do whatever.
exactly. So let's start with the first mushroom of the afternoon, the state mushroom, Agaricus julius. You know, this is common under spruce. It bruises yellow, has a shaggy stipe when it's young. It has a beautiful like cherry almond aroma when it's fresh. The problem is the bugs get to it before you do. And, it's rare to find a perfect one, but it is my favorite mushroom for the table in Agaricus in general. It is our state mushroom. A lot of us got together and we had a whole state mushroom committee of all the greatest mycological minds throughout the state. And this is what we chose out there. And, CMS should be proud to have Greg Sanchez and Alex Merriman, who spearheaded this operation. We all jumped on board and we made it happen together as a team. And this is the kind of stuff we can do together as clubs. We can join forces and we can make amazing things happen. And this got signed into law and I can't even believe it. I can't believe I was a part of it. So thank you, Greg. So I had to bring up Julius first because I thought it was appropriate, right? So I've learned some things about Julius that are different. I mean, when you see this, you kind of know. You see it and you go, oh, yeah, it's Julius. It's just, you see the color, the smell, everything, and it's pretty obvious generally. And so here's some younger versions of it. And what you'll notice is when it's young, it has a shaggy stipe, like I was saying. And typically, you see this cap color or this brown with this pattern, and you know right away, that's a Julius, right? But what I found through DNA sequencing many collections is that we also have lighter versions of it. And if you saw one of these, could you tell me it was a Julius in the field? I couldn't. So, I found this collection under spruce and I was like, man, I can't figure out which one this is. And I kept looking at it closely. And that's the whole problem with this Arvenses group. Once they're lighter like this, it's hard to tell the difference between them. And then I saw these. I had no idea this was a Julius. But once we found out from the DNA, I'm like, wait a minute, there's something going on here. And it's really the same thing as the button mushroom in the store. So you got your cremini and then you have your white button mushroom. This is the same species. But in the wild, there's variation too. So to me, these are like different strains of the same species. And there are a few that do that. So it's deceiving. And you can't always say, okay, it's always got this certain cap. Julius is not always like this or like this. It's also like this. And there are other species that are that way too. And we'll get into that next. So that's the interspecific variability. It's something I don't think it's well documented. And I think it's important to note when you're out there trying to find these. So the next one is Agaricus bisporus. This one took me a while to kind of nail down. And this picture is actually on straw manure, of which it's very fond of, I've noticed. I actually had a foray. We had a foray in Black Forest, Colorado. And I have a friend that lives there and he has some horses and I went to visit him after the foray. And I walked his property and then, I started finding mushrooms and I'm like, cool. And then I got into his horse manure pile that they were like pushing out of their barn with a straw. And there were Agaricus everywhere. And I went, dude, these are good edible mushrooms, and they would never eat them. They'd be afraid to eat these mushrooms off their horse manure pile. But what I found is that they are fond of the horse manure thing. They're not always found with it, but it's very common that our wild bisporus will be found with horse manure in the wild. I've also seen it at 9,500 feet in Cripple Creek, Colorado on horse, which is odd because usually it's not found at that elevation. But Agaricus bisporus it's extremely variable in its appearance. It generally has a brown cap like this. And then here's some more varieties of it. So this is a little bit lighter and they're in the button stage. And then here's some darker ones that were found at Colorado College on the lawn. And one thing I do want to say before that last one. So you'll see I scored it right here. I scored that to see the staining color. And that's one of the things that helped me identify it because bisporus stains a little bit orangish red. It's not always pronounced, but if you scratch a fresh specimen like that or you cut it, you'll often see that. That's one of the ways you can help identify it. So here's some more pictures of it. I have multiple pictures of this species because I think it's an important one. Whether or not this escaped cultivation or not, that's up for debate. We really don't know. Here's another way you can see in this button, you see that little orangish red right there, and that's what clued me into what this one was. And then here's another example of the variability in cap color. So these are both bisporus. This one was found at Boulder Park, Colorado Springs. This one was found actually right outside the Manitou Experimental Forest headquarters. I walked out the door and it was right underneath a pine tree. And then I smelled it. I ended up actually scratching it and I'm like, I think this is Bisporus. And it turns out it is. So these are just good examples of the difference in the wild, right? So you have, this is typically the color it is, typically brown like that. It's a little bit lighter, brown again, and then all of a sudden we have lighter versions. So I think this plays out with many more Agaricus, not just Julius and Bisporus. Chocolate brown is the common spore color. Yeah, there's really not a lot of variation as far as spore color goes with Agaricus. A lot of people go, oh yeah, it has a brown spore print. I'm like, yeah, we know, like that's pretty normal. Unlike other mushrooms that may have some more variability, it's more in the microscopy where you see the differences, the size, the shape, some other features. This is Agaricus subflocosus. This one for me is rarer to find, and it shows up in weird random places, and it's very closely related to Bisporus. So if you find something like this, you might think it's Bisporus. And they're very closely related. So I haven't quite nailed this one down entirely. It does exist here, and here's two different versions of it. The cap is a little bit different sometimes, but not always. I have some more pictures of this that make it look just like a Bisporus. And before we continue, I do want to say that Bisporus itself is typically found in the city, on lawns, all on the Front Range, but it's also found up under conifers sometimes more rarely. So I found a collection under spruce last year, and I've heard of it being under spruce, but that was the first time I actually found it. And I thought it was subflocosus by the way it looked. I don't have a picture of that here, but that's another thing to note. But this guy is found all over the place. So this subflocosus was found under pine up about 8,000 feet. This one was found down in the cottonwood riparian about 6,000 feet. And it doesn't show up very often, at least for me. I'm sure other people find it, but. And then here's our Campestris. I'm gonna start trying to move faster because we have a lot of material to go through. Campestris does occur here. We have 4 or 5 different versions of this Campestris, essentially. But this is the real deal Campestris. So this collection was sequenced from Divide, Colorado, and it matches type species of Campestris. So we have the real Campestris here. And that's been debatable over the years. A lot of people debate that, and they think we don't, but we do. And what I've noticed is that it's usually kind of mid-elevation. This is around 9,000 feet out in a pasture, so they like to be out where there's grass. And if you go out where it's open and it got really wet and there's been cattle grazing, they like that too. So that's the most common places to find it. That's why I have about 8 to 10K in here. But we have a few other ones that can be deceiving that you might think are Campestris and they're not. So here's one of those. This one is generally found in the city. If we would get some rain, you could probably go over to the park over here and find these. These are in the same group. They're very closely related. You'll see when you cut them in the button stage, they have a pink gill. So that's one of the features. We've called these Campestris forever, for years. These are actually Brandeli. They were called Argentius, but that was an illegitimate name. It wasn't published properly, so it ended up becoming Brandeli. It's also got a close relative called Andrewii in the east. But this you'll typically find from all along the Front Range, in the parks, in the grasses. It loves grass all the way to the east. But it also shows up in mountain towns like on lawns. So Telluride, I have one from a Telluride lawn that Andy Wilson gave me. So it can show up in other places, but it's from here east, and it likes grass. They don't really stain in this group. So the Campestris group is the main section of Agaricus, and they don't have a lot of staining like the other groups do. Good question. What's that? It is a smaller.
The DNA level, is that what you're?
There are subtle differences. I mean, it is a smaller Agaricus. It's also where you yes, the DNA is slightly different. Yes. What was that?
All only differences.
A lot of it's the same. I mean, depending, you could debate like the ring is a little different, but it really depends on the stage it's in. It's hard to find stuff with a perfect form, like these started drying out. These are really fresh. I was actually with Scott Ostuni when we found these in Colorado Springs. And I told him they were Brandoli, and he's like, "Are you sure?" And I'm like, "Yeah, I'm sure." And then we got them back and they aren't, because I've collected many of them. So I've gotten used to it. But this is our city Campestris. That's what that is. So here's another one that you might think is Campestris at high elevation. This is generally found over 10,000 feet, in my opinion. This picture is from Telluride, but we found it in the wet mountains too. And so you might find that and go, "Hey, I found it. I found Campestris." And most people would. But if you look at it really closely, you see it's got this tan color on the cap. See that? And it's usually found in the conifers. So this was found under spruce at like 11,000 feet, I think near Alta Lakes in Telluride. But we also found it down in the wet mountains, and it's showing up all over the state at high elevation. So this is becoming pretty common. And there are a couple different versions of it, but our subspecies is called Palladius. And this is another Campestris relative. I will get into some more Campestris relatives, but after we get towards the end, which are newer ones. So here's a Bitorchis. This is pretty common in our city. It's easily confused with Bernardii. It took me years to figure that out. But one of the things you can look at this picture, you can see it's got this double ring. They're short and stocky. It's got a double ring generally. If you get them in the right stage, you can kind of see it right there as well. Bernardii has more of like an enrolled cap margin. It also has like a stronger briny smell. And then if you were to cut them, they rarely stain like pinkish red, just a little bit, where Bernardii is more pronounced and more common and all over the place. But this one takes a while to learn, but that double ring is definitely one of the main features. And city habitat. So here's Bernardii. This is the most common Agaricus found, I believe, on the Front Range. It's everywhere. It's in every city park. I find that it often prefers where the dead grass is. So if you're walking, through a park or somewhere where the grass starts to die, oftentimes you'll find them in big rings in and where the grass is dead. And what they're doing is they're decomposing that dead organic material, and they're recycling it and turning the soil back into fertile soil. Another thing about this one, it'll start to turn a little bit tan on the top, and it has this tendency to crack. Other Agaricus can crack like this, but this one's probably one of the most common that cracks like that. And then it's got a cottony veil when it opens. And after it opens, the cap margin will be very enrolled, and they're usually really robust, and they're not very tall. But you can see the red staining in here. So I wish we had one today, because this Agaricus smells different than any other Agaricus that we have. And if you get that smell, if you can smell pretty good, you'll cut that and smell it, and you'll know immediately what it is. Here's another relative, which is a little more rare. It's called Pilosporus. And the first time I found it was in my yard. And this picture is also from my yard. I found it again. It's another red stainer. It's similar to Bernardii in smell, but a little bit less, in my opinion. And then it's also a red stainer. And this is one of the first described Agaricus from Colorado. So it was described 122 years ago by Charles Horton Peck, which I couldn't believe when I found that out. I was like, oh my God, there's an Agaricus I didn't know about that's 122 years old from our state. And so I'd like to find more of that. So now we'll get into our yellow stainer. This is always called Xanthodermis. So if you open any of these books before we had, a lot of DNA sequences, they call it Xanthodermis. And what we found from sequencing many, many collections is that it's a close relative. The DNA is very, very close. But this particular one was described from New Mexico, and it's found all up and down the Front Range. And this is the one that you want to avoid. And this is the one that everybody says stay away from. You don't have to stay away from all the yellow stainers, but what matters is where it stains yellow. And so if you look at these pictures, you'll see it's in the context of the stipe at the very base of it, and it's bright yellow. It's really bright. I can just pick one of these up and smell it and know right away what it is. They have kind of a chemical smell, and it's unpleasant. Another thing that you'll notice about these is you look at this one, I have more pictures coming, but there's a darker cap, depressed center. So the cap's often it forms this dark center to it. When they're in the button stage, they're kind of more like a deformed marshmallow. And when they're fresh, they always have a bright yellow staining right at the base of the stipe. They also have a thick felt-like ring right here, and they have a unique stipe base. So if you look at the base of this, they're very unique in the shape that they are. And then you can see here it's staining yellow. They do have a variable appearance depending on the conditions. So you look at this one, it's very tall. These ones are very short, but these are the same species. They still have the felt-like ring that's really thick. They have really bright yellow staining at the very base. And then you can see this darker depressed center. They also form a cap shape that's kind of like bell shaped. So here's another collection that looks like kind of like a bell almost. It's very unique to the species. And you can see a little bit of yellow staining right down here. This is my favorite picture of it. And it was protected. It wasn't out in the sun. This is at near Bear Creek Nature Center in Colorado Springs before we had a lecture. I was walking around and I found this and I was like, really cool. You can see the thick ring there and then you can see the yellow staining right there. But you see, they start forming the darker depressed center and they have this unique cap shape to them. So there are a lot of ways you can tell what this is. Yeah, and a lot of Agaricus have a marshmallow shape, but we call it a deformed marshmallow. So kind of more like this guy is really common. But if you know where to look for the yellow, you know what it smells like, and then you start putting these other subtle differences in, it's really obvious. And so we don't really find these in the mountains too much. These are more of like front range, maybe up into the foothills a little bit. And, you know, most people want to tell the difference between Agaricus arvensis and Malangellus. And so I'm still calling it the yellow stainer, but, you know, when I was in Telluride last year, I think I was talking to Ed, who's back there. And, you know, I do speak Spanish and I didn't even think about it. And he's like, oh yeah, Malangellus, that means bad angel. And I'm like, very good. Why didn't I think about that? Thank you, Ed. So here's another one. This is a new one. And I want to thank Catherine Williamson and Darcy Rose for these pictures that they're letting me use because I don't have any pictures of this species, but this is another one in that same section that is a yellow stainer, and you can really see it in Catherine's pictures right here. This is another section Xanthodramati agaricus. This is another poisonous toxic agaricus. And this was recently discovered in Colorado, and it's a southwestern species. So these were from Ridgeway, so, you know, southwestern part of the state, and these are actually from New Mexico. It's more prevalent like in New Mexico and Utah and stuff like that, but this is a new one for us. And I don't know if Tom's here. I don't think he's here. Yes. Yeah, it's bright yellow in the base of the stipe, and that's what is typical in this section. And that's one of the ways you can tell it's in that section right away. Usually immediately, like right afterwards, yeah. And it's usually at the very base. So I've never actually found it, but we have two collections now from Colorado, and you can see this one's got like a darker cap, turns a little grayish and kind of brown. And what I wanted to say, Tom, if you're listening, this is very closely related to Agaricus deodorophensis from California. And he was asking one time if we have deodorophensis here, and I don't think we do, but this is probably the closest thing to it, and he may have found that. So we're going to get into the section Arvensis woodland agaricus. So if you look at any of the old books, you're going to see they're going to call this Sylvicula. It's my understanding that Sylvicula is a western species from California that's related. It looks very similar, but we have several different ones in Colorado, and I don't believe we have Sylvicula here because we don't have any DNA data to verify that, and we have tons and tons of collections now. So I believe what we're calling Sylvicula is probably Didymus or Gemellulatus, probably Simensis too. Here's the first one. This is the type species of the group. This is Agaricus arvensis. This is more of a city mushroom, so this doesn't really occur too much in the woods, but it can get up into the ponderosa pine sometimes. June is a good month for it. They found on lawns, and they can stain a little bit yellow, but usually it's like on the edges, you know, the cap and things like that. They often grow in fairy rings, which you see right here. That picture's from Woodland Park, Colorado last year. I was waiting for somebody and I think it was at the church over there. I saw a bunch of Agaricus in this field and I jumped the wall and I ran over there and I got this picture and then I got this fairy ring picture. And I smelled it and I'm like, oh, cool. This is a higher elevation Arvensis because usually, usually it's found, on the Front Range in the cities and the parks in Denver and Colorado Springs and stuff like that. But it can be found in mountain towns where there are lawns. So it has a pleasant odor. It'll never stain really bright yellow at the base of the stipe right here, like these guys do. It has a pleasant smell. And actually, it's very good to eat. And most people confuse this with either the other yellow stainer or Campestris, but this is larger than Campestris. And if you look, I don't know if I have a picture of it. When it's in the button stage, it's not quite as bright pink. It doesn't have that bright pink. So if you were to cut like a button, it wouldn't be bright pink. It actually start out like white into gray, as does everything in this section. Here's some pictures of Agaricus didymus. These are 100% matched to the holotype, both of these. And it's really tricky to identify didymus and gemellulatus because they look identical. The DNA is so close that some of the ones that are in between, you cannot tell. So these are 100% matched to the holotype. And so I'm able to say these are didymus match, but you have stuff that's like a base pair or two different and it's all in between. And when we run trees on this, they all kind of group in the same place. So, they're either very closely related or they're pretty much the same thing. And there is some hybridization going on as well. And we have a little bit of evidence of that, I believe. And there's also a secotioid form of this. And it's not Inopertus like we thought. So there's some variation. This is 100% matched to Agaricus gemellulatus. Another thing that we know is that Agaricus inopertus is a secotioid, and there's one called Mesocarpus, which I can't find, but these all share the same DNA sequence. So really, the only way to tell the difference between gemellulatus and inopertus is that they actually open. But we have some, I think in the DBG, some inopertus, but I don't think we have sequence data on that. So we don't know, honestly, we don't know if those are really inopertus or not. And that's why I've taken it kind of out of my list of species because until we can prove that, I'm not sure if it is really here. It's very prevalent in California and Arizona and stuff like that. So here's a secotioid version that's slightly different. I initially was like, oh, cool, that's inopertus. We finally found it. Thank you, Hamilton. This is great. And then, the DNA is slightly different. So we don't have a name for this. This probably should have a provisional name associated to it, but more work needs to be done and we need more collections. So we really need to find more of these, Dimitri. But this is a really cool mushroom. It doesn't really have a name. It's slightly different. It's in that group. We know there's hybridization going on. So it's a really tricky thing to figure out. Yes. I think we should be looking in the southwestern part of the state or in parts of the state that are drier. This was found on Rampart Range, which is shocking to me because I've been hunting mushrooms there for 20 years and I've never seen it. But Hamilton comes to town and he finds it. So it's really awesome. And the other thing I should say is that it's my understanding these are adapted to dry climates to prevent desiccation so they can still, disperse their spores later. So it's a way they're protecting themselves from the dry as we go into more drought. Here's Semensis, and it's very similar to the Didymus gemellulatus. If you found it, you probably couldn't tell the difference. This one, though, can turn a little more golden orange in the color, but Sandianus also does that too. And so unless you're going to DNA sequence this, you're not going to really know which one's Semensis or gemellulatus or Didymus or any of that. It's very difficult. They're identical. They're very closely related. But one thing I will say is that the Didymus gemellulatus group generally shows up under spruce, sometimes under pines, where Semensis, the majority of the time I find it, it's under ponderosa pine for some reason. It does not mean it can't occur under spruce, but it seems to me that it occurs at the lower elevations where the Didymus group kind of occurs a little higher, but they do overlap. This is Agaricus sandianus, as described from New Mexico. We have I believe 3 or 4 collections now in Colorado. And it's another one that's hard to discern, and I think we need more collections to see, like, does it have a bulbous type base? This one doesn't, but that can change as they mature and they grow. This one's from Telluride, from I think last year actually. And that's another one. And then here's another one. So this one was originally called RWK 2283. It's named after Rick Kerrigan. It's in his Agaricus book I have over here. And it's a little bit smaller. It's similar to Moranii. And what I've noticed, or what we noticed, is that the stipe base kind of has this certain, like, a saucer shape to it. And so it's a little bit smaller, and it is a bulb, but it's not as bulbous as some of the other ones. So, if you find something that has, like, kind of almost like a flying saucer to it at the bottom, it probably is the species. And one of the things I'm trying to do is figure out the morphological differences between all these different Agaricus that look identical. And some of these are almost impossible to tell the difference. This one has a few different characteristics that, you could almost narrow it down. We know this occurred in our state for a long time. This is Colorado's version of Agaricus moranii. It's more prevalent out west. In fact, the ones that are out west are a little bit larger. They could even have more cap color to them. But the ones that I found in Colorado are very short compared to the others. You can see they got a little bit of a bulbous type base, but you could confuse them with other ones in this section as well. They all have yellowing. They're found in the woods. These were from about around 9,000 feet elevation. These are from Florissant, Colorado. And it took me a long time to actually find this. I found a few of them now, but it does happen. Okay, so Nan Augustus was a surprise. We found it on Palmer Park lawn, and it's related to Julius and Augustus, but this is the eastern prince. So this is very prevalent from the Front Range east all the way back east. It was growing with some type of imported conifer. And I think that's how it got here, but I'm not sure. We found 2 collections of it now, and the other one was under some junipers actually, and it's all cracked, so it looks a little different, but we do have prints in Colorado. This is Agaricus amicosus. This is always a treat to find when you're looking for boletes and you can't find them because you're under spruce, and it loves spruce. And actually, I think Jennifer Bell found this when we were with David Aurora trying to find Agaricus julius in 2021. And she found these, and then we were looking at them, and I remember him having them both in his hand, kind of just looking at them and thinking, and I said, they're different. And he's like, yeah. And I said, those are amicosus. And he said, yeah. And then that was the end of the conversation, but he set these up, and I took a picture before he, took his pictures. So thank you, David Aurora, for setting those up for me. This is a fun one. This is Agaricus bonensii. And this one took me a long time to track down. I found it over the years, but the problem was when I would send it for DNA, it would fail. And we always thought this was Sylvaticus back in the day. So, like, if you found something in the woods like this, a lot of people would go, oh yeah, it's Sylvaticus, because it's got this blood red staining. It's very, very bright. It's one of the brightest red stainers in the forest. We call it the bleeder. And this is common. I found this one under Douglas fir and then this under spruce. So it starts out very white. And actually, I rubbed these too much before I was trying to take the picture. I was trying to brush all the dirt off them, and they started turning more red. And then later on, found this collection, and you can see they start to turn like a brown color, brownish red on the cap. There are a couple other species that are similar, but this one's probably the most common. So if you find a white Agaricus that's really bright red staining in the earlier stages, it's probably this species right here. This is Agaricus sylvaticus, subspecies occidentalis, and this is one that we do have that's Sylvaticus. These are really, really skinny, and they don't typically look like this from other states. They're usually a little bit thicker, but depending on the environment and if they stretch or whatnot and how dry it is, you never know. So we had a foray at Palmer Park in Colorado Springs, and Chef Beth actually found these and gave them to Alan. And then when I came down from the hill, he's sitting there taking pictures, and I'm like, hey, what is that? And this is a new find. So we need to document this more. This is Agaricus frisianus. This is one of the mushrooms that started this project because I would find this little mini Agaricus and I knew it was an Agaricus. I had no idea what it was. I really feel like in the DBG, there are a bunch of these called diminutivus because it represents diminutivus, which is from the West Coast. But we have frisianus. Oftentimes, it'll have this little pattern here and it'll blush. And it's really long generally. But it can easily be confused with Agaricus carraganii, which it looks very similar and it's very closely related. And it's easy to misidea it for that reason. But these are generally found at lower elevation in the ponderosa pine and the gambel oak. And so this is one of the first Agaricus I was trying to identify. And when I had access to DNA sequencing and we did a DNA sequencing workshop, this was one of my first 12 collections, or a different one, but this is one of the ones I was trying to find because I didn't know what this was. And I'm just a guy trying to figure out how to identify these mushrooms. That's really where this started. I have an obsession for identifying mushrooms. People ask me what they are. I want to know what they are. So that's how a lot of this started, was from this very mushroom right here. Here's another one in that same, mini Agaricus section. It's one of the larger ones. This was a rarity that was found when we were doing a foray with NPR a few years ago. And it showed up at my house on the counter. And I'm like, well, what's this doing here? And I started asking questions about it. And then we found out this was Agaricus jacobii, or Jacobi, sorry, which has only been found in Washington and then, Spain before that. So I believe it was described from Spain. And I was like, what's this Agaricus doing here? This is the only known collection of this in Colorado.
Get on your kitchen table.
Alan Ronkerpiller brought it to my house because somebody gave it to him. And I was like, dude, why is this sitting here? What is this? And then I started trying to figure it out. This is another one in that section, Agaricus carraganii. This took me a while to figure out. They're usually shorter than this, but they're extremely variable. They always have this similar cap pattern on them. And oftentimes, you can't really see the ring on this one, but oftentimes the ring is very unique. They do have a little bit of yellowing. These are very, very common. And last year when we had our joint foray with CMS, I saw a ton of them, more than ever. And that's where I got these pictures from, was from our joint foray last year on Rampart. Agaricus altipes, this shows up randomly. I only found one collection of this. Actually two, sorry. I have a cap from Telluride as well. I really want to find a good collection of this because I want to get good photos of it. These were really soggy when I found them. And, I was sitting there trying to figure out, and I couldn't figure out for the life of me. And then once we figured out these were altipes, I was really excited because then this is another one that was discovered here that we never knew existed in Colorado. This is Agaricus cyanodermis. This one's pretty common, but it's extremely variable in appearance. And this shows up, this is another woodland Agaricus that's not in section arvenses. This is one that shows up under spruce at high elevation. This is one that also grows over tree line. It can be variable in appearance, but it is in the main section of Agaricus, which is related to Campestris. It's just a bigger, taller woodland version. And if you notice where it's sliced right here, the pink gills. So if you were slicing something like Didymus or Semensis at this stage, they would probably be like white or gray at this stage. And then also the cap color is a little bit grayish, and it can be a little reddish, but mostly gray and whites. But if you find this and you're able to slice it early and you see there's pink gills, it's very likely this species. The smell is also not like almondy like some of the other ones, but it can be deceiving. This is kind of a weird, funky species that sometimes can yellow, and it will trick you. The other thing you can see is it's got this enrolled cap margin, and they can get fairly large. I mean, they get really big. It has a close relative called Agaricus rhizocephalus. This one is a little bit shorter, but you can see it has more of a reddish brown to it, but it's variable depending on the collection. And you can see here it's got the pink gills early in the stage. So right away when I found this, I knew this wasn't, your typical woodland Agaricus because of some of the features it was showing. Also the smell is obviously a big deal. It took me a while to track this one down, but these two, are closely related. And we do have one that's in between these two species, and it looks like it's a hybrid of these two, but more work needs to be done. And it could also be a new species, but did not include that in here. But this one's a really cool one. And yeah. This one I found in my yard one year, and now it shows up every year except for this year because it's too dry. It's a very large Agaricus. It's a red stainer, and you can see a little bit right here. There's a little bit of red. It's not super red. This is the only known one in this section that does not grow near trees. There are only 2 in this section in Colorado, and this is the only one that we've been able to actually find. They call it Agaricus species AG because it was temporarily named after these 2 guys from Wyoming who plan to formally describe it in the future. And that's according to Rick Kerrigan. In the Agaricus of North America book, it's in there, and he talks about that. And he said he's leaving it alone so they can describe it, and they still haven't described it. But they need more collections. They need more pictures. So now I have like 3 collections of this. I should probably send some to them, to those guys, if I know, can find them. So it's called this Agaricus species AG. It's provisional. So we're getting into some new stuff here. So anything you see up here, it says Agaricus species and it has a number. This is a temp code. So the reason why we have a temp code is because this doesn't have a name and it needs to be described. This actually came from Denver. We drove past the park earlier where it occurs. If you found it, you might think it's a torque because it's very closely related. They look very similar. But if you notice, the ring is really low on the stipe on these. And we need more collections of it, but Scott Ostuni discovered it. Nothing else matched it, so we gave it the temp code, Colorado 01. This is the first new Agaricus with a temp code, and this is likely a new species that needs to be described. Here's Agaricus species C002. This is a very small Agaricus that grows in my yard, but it hasn't shown up for 3 years. And I really want to get good pictures of it, but the problem is once it shows up, it's out in the sun and it gets fried really easy. So they dry out really fast. They're really small yellow stainer. I'm not aware of this being anywhere else in the state, but it has to be. We have to find more of these. So this is a new one as well. It would be nice to get more collections of this so it can be described. This one is called C003. This is another Campestris relative. There's only one of these that's been found. It's only this one. And it's very similar to Brandalize. So it's possible that people have found this, but we haven't sequenced any more collections. This is the only one that has a sequence. And we need to find more of these. But again, this is just another Campestris, essentially, variety, complex. This is another version of Campestris that is C005. I asked Darcy to use her photo because she's the only one with the photo. And this is a high desert Agaricus in the Campestris group. It was found outside of Montrose, Colorado, and as you see, it's in the cactus. It's in a super dry area. So when you see this, if you saw that, you'd be like, oh, that's Campestris, but it's a little different. So this is a brand new one that I think is really cool. Hopefully we can find more of those, but I don't frequent the Western Slope high desert areas too often. So that's why we need, other people collecting, people like Darcy, who's collected tons and tons of stuff and provided a ton of data for science. I put Agaricus abruptibulbus in here because Ian Fireman asked me one day, he's like, hey, do we have abruptibulbus in Colorado? And I was like, yeah, I don't think so. And the reason why I said that is because we didn't have any evidence of it. So what happened was Catherine Williamson, my friend from the New Mexico Mycological Society, found it in northern New Mexico. And I helped her validate the sequence. And when I did that, I was like, oh my God, you found something that, we've never found. And we talk about abruptibulbus. It was originally, the original collections from New York, and it does occur like in Arizona and other places. And so this is the very first one that has occurred north of Santa Fe, New Mexico. And those are the Sangre de Cristo Mountains. And those mountains go all the way up into Colorado. And so I believe that's a very good possibility that this exists in the southern mountains of Colorado. So I put this here to answer that question for Ian. I think it very likely exists here. I put this up here because This has been taken off the CMS website. This is how I started learning Agaricus years ago. This maybe 20 years ago, I pulled this off your website. And Ellen Jacobson, she wrote this. And I started reading through this, even though this is very out of date, there's still a lot of good stuff in here. This is what really got me started. I started reading this and I go, oh my God, this makes sense. I can smell, I can do this, I can do that. And so some of these tips and pointers is really what got me started a very long time ago. And I was just trying to figure out what the differences were. So I did make a copy of this. I found this on the internet, I don't know, a year or two ago. And I actually saved the page on my computer, thankfully, because it's gone. It's not on the CMS website. It's not on the Angel Fire site where I found it before. And thankfully I saved it because I think this is a fantastic thing, even though it's out of date. And, you know, CMS had this on their website and it was you guys that provided that and got me really a head start. So thank you, Ellen Jacobson. And these are all of my references. I can't believe we're here. So thank you. Oops. Okay, I'm going to open up questions. Who has questions? Okay. Okay, questions for Agaricus bernardii? The salt part or the bernardii part? That I don't know, but I do know it's been called the salt-loving agaricus forever, and it's found along paths and places where they use de-icer and things like that. I don't know how true that really is. I
James?
mean, it does smell briny, like in a briny smell, so that could be part of it, but I'm not sure about the history of that.
James,
One sec. They're probably absorbing all sorts of stuff that's in the city like that, pollutants.
James,
Yes.
when you get a question from your live audience, could you repeat the question? We can't hear it at all.
Okay, I'll try to do that.
We can hear your answer, but we can't hear the question.
Gotcha. Any more questions? Done. Wow. How about the Zoom people?
I have a question about the spores of different agaricus. When you're looking in the microscope, are they generally all pretty similar, or is there quite a bit of variability?
A lot of them are very similar, but some of them have some of them will have an apiculus, so they have like a little protruding thing sticking out, and some of them won't. Some of them will be a little bit more rounded than others. So, the length to width ratio will be different, which is also called the Q value. Some of them have like a thicker, like around the outside of the spore, it looks like the edge is like a thicker, I'm not thinking of the proper word for it. But they do look slightly different. A lot of them look the same, but I do believe that they all look a little bit different.
The cell wall can be different.
That's it, the cell wall. Alexis? Mm-hmm. Yeah, I think, okay, so the question is, Alexis has a question of the section Nigrobursentes, which is, we have two agaricus in that section in Colorado, only one that we found. It does mean dark or black, but those are actually red staining. I'm not sure why they called it that. Well, I think they actually have like varying colors depending on the species and other places. So like Liliceps is one of them. Agaricus liliceps is known from California under cypress, and it's a close relative, but there are all different kind of like reactions and stuff going on in that group. So some of them can have purples and reds and probably darker colors is my understanding. And that's probably why they call it that. I don't know a whole lot about that section other than we have Agaricus species AG, up and down the Front Range in the plains. It's like a Midwestern species. And then we have another one called RWK 1583, which is in Kerrigan's book, but we need to find it. Sure. Question is anybody cultivating Agaricus other than the grocery store variety? I don't know anyone in particular. I've tried to put Julius on agar and failed. I put Bisporus on agar and got it to grow, and then it stunted and it grows really, really slow. So I think you need to have the right medium to do it properly. I did want to get a Bisporus going because I wanted to put a bed in my garden. But Julius, since it's our state mushroom, there will be a lot of attempts to, clone a Julius to try to cultivate it. And we've been trying to do that for a couple of years now. And I was actually talking to Graham Steinruck recently about growing Agaricus, and he was talking about how slow they grow. And then he told me about a certain type of agar that may have the proper nutrients in it, can actually grow better. Yes. I'm trying to go to it. Okay. I'm trying to hear you too. Okay, so the question is, why are we calling this the main section Agaricus up here? I do believe that was one of the first, and the main, I believe the main and Arvenses split first. And if you look at this slide, you can see right here, about 30 million years ago, it shows they split. I believe these are the two oldest sections, yes. Correct. Although this one says 33, but we don't have any in this section in Colorado that I'm aware of. We have all these top one, well, not all these, but we have some of the ones that are in the top, and that's what this slide is about. This slide shows you all of the different sections that we do have that are verified. Okay. Question is, can I explain more in depth why certain Agaricus species are more prevalent to growing in lawns or in the city? That I don't actually know. I mean, for me, they're just decomposers, so they're decomposing the grass. And I think maybe it's that they prefer the grass, but some of these actually prefer certain trees. But most of the city Agaricus are just out on the lawns and whatnot, and they like that environment. I don't have a full answer for why that is, other than they're decomposers. Yes. One more time. So your question is about them having relationships with certain trees, but not being mycorrhizal. That's correct. They're not mycorrhizal. They're primarily just decomposers, and they do have relationships with certain trees, and we don't know why. There could be, some more information on that. I don't particularly have it. I just know that, when you find Agaricus julius, it's under a spruce tree, and they have some sort of relationship with that tree. But they're also found under Douglas fir or ponderosa pine at times, more rarely. But certain Agaricus, especially the ones up here, except for Arvensis, the type 1, all of these are found under conifers. That's what they like. But you would never find any of these in the city except for this one right here, the top one. We don't know what the relationship is, and that's a difficult thing to probably figure out. But there could be information on that I'm unaware of. Yes. Yes. So the question is, are there places that I would caution against eating agaricus because of heavy metals and stuff they would uptake? Right? Yes, I would caution against eating anything in the city that's on a lawn that's been fertilized next to a road where there's a lot of cars driving by. And a good example of that would be, um, Agaricus bernardii, because it's very commonly in those areas. Another place that I would be cautious of are these woodland agaricus in places where there are mine tailings. So outside of old mines where there's a lot of, heavy metal toxicity, that would be a place for caution as well. And we don't know a lot about that. And, we take that risk with any edible mushroom that we're eating out of the woods that's anywhere near a mine or an old gold or silver mine or whatnot, because places like Leadville, we know they're polluted, right? Oops. Where did it go? So yeah, I would caution against anywhere that has, a bunch of mining activity from the past or close to it, or just stuff in the city in general. But, I wouldn't eat any of these that grow off the lawn unless they were in my backyard, probably. So like if I went to where I found Bisporus at Colorado College in Colorado Springs, they put sod down, they put weed and feed. There's cars driving by. Actually, I did eat a Bisporus off their lawn one time because I wanted to, but I don't typically do that. And I know people who don't even care. They're like, oh, I'll just eat whatever. But you are taking a risk by doing that. Okay,
I have another question.
go.
I love all the different color variations from when you scrape on the mushrooms, like pink or red or orange and yellow or dark red. I think that's fascinating, and I just wonder more about that. And in relation to using potassium hydroxide, do you even need KOH or can you just scrape on them?
I mean, it's a good idea to get a KOH reaction, but at this point, we pretty much know, if it's in the Arvensi section and use KOH, you're going to get a yellow reaction. Same thing with the xanthodermati, but some of these don't really have a reaction. It's a way to identify it. I don't always have KOH. I probably should. I don't think it's necessary because you can smell and you can usually see the staining without it. But if you want to know for sure that's a yellow stainer, that's a for sure way to know.
Do you know if any of them might stain yellow with KOH, but not stain yellow with scraping it?
No,
So
unless
they're
it's like aged.
pretty
think
consistent.
Yeah, anything that stains yellow is probably going to stain yellow with KOH. So it's probably these three sections on the right of the screen here.
Mhm.
Arvenses, Minoris, and Xanthodermati these probably all will stain yellow in some fashion or with KOH for sure.
Okay. And I did just want to say thank you for all your help on iNaturalist with identifying so many mushrooms, all kinds of mushrooms, but particularly the agaricus. And it's really great you share so much information and knowledge, and thank you very much for that.
Thank you, Catherine. Yes. They don't necessarily cook out, but I got it right here. This is what's making people, sick when they eat that, the hydroquinone. That's the toxin that's found in those. Some people say they can eat them, but I don't think I would try. I mean, it's not going to hurt you that bad, but you probably don't want to throw up or have a stomach ache. Honestly, I think they don't smell good. I mean, any edible mushroom that I find that doesn't agree with my smell, I don't want to eat it, right? Okay. Anyone else? Oh, they keep coming. None that we're aware of here. I do believe there is one that is really toxic somewhere else in the world, but I don't have a lot of information on that. None of these Agaricus that we have that we're aware of will, kill you per se. But I mean, do know that even in these other sections, like anyone could be allergic to something different too. So like most people can probably eat all of these, but then some people might, have a reaction to it, an allergic reaction. So a good example of that is something that's not on here. So we have one that we used to call Agaricus abolituscens, which would be in this group right here. But the reason why it's not on here is because we have no DNA verified proof of that, or at least I don't. So I don't believe that it exists here, but if you read in some of the older books, it says, oh yeah, this could cause gastrointestinal upsets. And for the most part, I believe that's false because of the section that it's in. However, different people could react differently to it. It's not very likely. Is that it? Thank you. Thank you, everybody. I don't know when I'll do this again. I was asked to be here, so I'm here. So thank you.
James. Thank you.